So previously we've discussed skeletal muscle contraction just a little bit in terms of the microscopic anatomy and relating it to the sarcomeres within the myofibrils within muscle cells. But let's take a closer look now at skeletal muscle contraction. And so we'll start with this picture and remind ourselves that we have the muscle. Within the muscle we have fascicles, which then within the fascicles we have muscle fibers, muscle cells. But there's a connection to the nervous system which is ultimately going to signal for the contraction of muscles. And so we haven't got to the nervous system yet, but we can think about the brain and the spinal cord and then talk about motor neurons. We're just showing one in this image here, bring signals ultimately out to the muscle fibers, the cells themselves. And so in this image, we're showing a motor neuron that comes down and branches to connect to multiple muscle fibers at motor units. If we were to zoom in on the motor units, we would see that the end of the neuron is not quite connected to the muscle cell itself, but we can then discuss the process of how we go from the nervous impulse, which we'll just say is an electrical signal for now, that's traveling down the motor neuron, to actually trigger contraction within the muscle cell. And so we'll take a look here at this image at the first step where the electrical signal travels down the motor neuron, and then it gets to the end of the motor neuron at the motor unit, and the nervous signal actually triggers the release of the neurotransmitter acetylcholine from vesicles in which it was stored until that signal came down the motor neuron. The acetylcholine then diffuses across the neuromuscular junction. Some of these terms seem fairly complex, but if we go back to neurotransmitter, the acetylcholine is the name of the chemical, and neurotransmitter literally means to transmit the nervous signal. And it transmits that by diffusing across this neuromuscular junction. Neuro referring to the neuron, and muscular referring to the muscle on the other side. So it is the junction between the neuron and the muscle. And so the acetylcholine diffuses across the neuromuscular junction, and then it binds to receptor sites. And then when it binds to those receptor sites, it causes a stimulation and causes an electrical impulse to then actually form on the muscle cell itself. And so the action potential then starts to be transmitted all across the plasma membrane or the sarcolemma of the muscle cells, and it travels all along the surface, but then down through the T-tubular system, as we mentioned earlier on in this section. The action potential travels down the T-tubules, and as you'll recall, and the image is here to show it, that the sarcoplasmic reticulum is butted up against the T-tubules. The sarcoplasmic reticulum is filled with calcium, but as the action potential travels down the T-tubule, it causes the sarcoplasmic reticulum to release that calcium into the cytoplasm. As the calcium goes into the cytoplasm, the cell is filled with myofibrils, and so the calcium then allows the actin and myosin to form the cross bridges that we saw earlier on in the image down on the right, allowing for the contraction of the sarcomere. But let's take a closer look as to exactly how that happens. If we were to zoom in on one strand of the thin filament, the actin in the middle, and then we looked at the myosin head down here on the left, we can see that on the thin filament there are actually these myosin binding sites where the myosin heads want to attach, but they are covered over via tropomyosin, another protein. The tropomyosin has troponin attached to it, which are shown at the top of the image here. And so under relaxed conditions, the troponin is bound to the tropomyosin, and the tropomyosin covers over the active sites of the actin, the myosin binding sites, and the myosin heads cannot attach to it. But when calcium comes in, notice what has happened. The calcium binds to the troponin, which then causes a change in shape of those proteins such that the tropomyosin uncovers or is removed off of the myosin binding sites. Then the myosin binding heads can actually attach to the myosin binding sites and shift in or pull the actin in, allowing for contraction of the sarcomere. And so that's what's being shown On the bottom here in the images, many myosin heads are binding to actin within the sarcomere, allowing for contraction. But here we've been able to zoom in on the process of how that occurs. So, as is stated on the left side here, this continues as long as calcium is available to the actin and myosin. For our muscles to relax, we actually pump calcium back into the sarcoplasmic reticulum. We have an active pump that does that. Calcium, therefore, is released from the troponin as more and more calcium is pumped back into the sarcoplasmic reticulum. And so the calcium will be released from the troponin. The tropomyosin will then recover the active sites, sort of knocking the myosin heads off of the active sites, allowing the muscle to relax. And so we will go from the contracted sarcomeres to the relaxed sarcomeres. In addition to the calcium being pumped back into the sarcoplasmic reticulum to allow for relaxation, we need to take a step back to the motor unit here and say, well, how do we get rid of that acetylcholine? So another step that occurs is that acetylcholine esterase, which is a long term here, but what it's really referring to, that ending ase, is referring to it being an enzyme that breaks down the ester bond, a type of bond we haven't looked at in this class, of acetylcholine to the receptors. And so it breaks down the acetylcholine and it releases it from the receptor sites. Therefore, the signal does not continue from the neuron down to the muscle because the acetylcholine is released as it's broken down from the receptor sites. Earlier in the course, we talked about energy sources, ATP, and how it's produced via glycolysis and in the citric acid Krebs cycle within the mitochondria of the cell and so on. So now we want to connect that content, that understanding, to describe the energy sources required for muscle contraction. Ultimately, ATP is the major energy source for muscle contraction, just like it's the major energy source for anything that goes on within our body. We make it through the same processes in muscle as we went through earlier in the class. But a little reminder, and zoom in on ATP here, that ATP is made up of adenosine and then it has 3 phosphate groups. Because ATP stands for adenosine triphosphate, there are 3 phosphate groups, triphosphate, with high energy bonds in between them. And so we can take ATP and we can break off one of those phosphate groups, and in releasing that phosphate group, we release some of the energy that's stored within that bond. And it allows for us to produce cellular processes, for example, like muscle contraction. In order to make ATP, we take ADP, adenosine diphosphate, 2 phosphates, plus an additional phosphate, and we undergo nutrient catabolism. In other words, we undergo glycolysis and the Krebs cycle, and we take the energy that's released from those the breakdown of nutrients, and we add that high energy bond to the ADP, forming ATP. Another chemical that's specific to muscles is creatine phosphate. And so we don't need to look at the exact structure of creatine, but creatine can be bound to phosphate and there will be a high energy bond within there. And so creatine phosphate, through processes that we haven't looked at yet, but through nutrient catabolism, we can take a creatine and we can take a phosphate and we can add it to the creatine and form that high energy bond. And so this is one way that our muscles store energy. And so the creatine phosphate cannot be used directly in order to allow for muscle contraction, but we can store creatine phosphate so that later on when we need energy, and we need energy fast, we can actually break down creatine phosphate. So release the energy from that high energy bond between the creatine and the phosphate. And then we can use that energy to reform ATP out of ADP. And this is one way that our muscles can make energy very quickly. So if we were undergoing some activity, let's say like lifting weights in the gym, where we need energy very quickly when we're all of a sudden lifting some weights, we can actually use creatine phosphate to produce ATP without having to produce the ATP through the glycolysis and Krebs cycle, which takes a little bit longer to do that. ultimately, after the workout, in that case, we will need to restore the creatine phosphate, and so glycolysis in the Krebs cycle will continue to work afterwards. But the creatine phosphate gives us this very quick form of energy, not to cause contraction of muscles directly, but to be able to quickly form ATP out of the ADP and the phosphate. Okay, so let's take a look at the effects of exercise on muscles and the overall body. Some of these processes we're going to look at are processes that you're very familiar with already, but we want to take a look at the technical terms for them. So the first, as an example, is hypertrophy. So if we break down this term, hyper means high, and trophy is referring to size. So that is the definition of hypertrophy, an increase in size due to use of muscles. And so if I have an inactive muscle here as an example, if I were to start going to the gym, my muscles would become more active and ultimately undergo hypertrophy or increase in size. This is not all about working out in the gym, but you might have somebody that you work with who, needs to activate and develop their muscles because they've had an injury, and they want to undergo the process of hypertrophy to get their muscles back to a strong and stable form. It's not always positive, however. So, for example, in cardiac muscle, as your heart muscle starts to hypertrophy, it can actually cause issues like increased blood pressure because there's less space in your heart for blood actually to flow within the cavities that are found within your heart that we'll look at later on in the course. And so this is not always a positive thing. The opposite of hypertrophy is atrophy. And so the prefix a means no, and trophy means size. And so it is a decrease in size due to lack of use. So you might see this in a patient who's had an injury. As I was mentioning, you might have them recovering from the injury and try to develop muscle, but it's because you've seen the muscle atrophy due to an injury where they're not able to use the muscle regularly. And so it would be the exact opposite of these images, where the muscle that is healthy and strong goes from active due to injury to inactive, and the size decreases. Another term that we should become familiar with is contracture. And this is the abnormal formation of fibrous tissue due to immobilization. Perhaps somebody has been immobilized due to an injury for a long time, and then their muscles ultimately undergo some fibrous tissue development within the muscle. And so instead of the muscle being able to be contracted and then relax, we end up with this sort of indefinite contraction or contracture, where this fibrous tissue has formed and is causing a tightness of the muscle, and so that the muscle is seemingly contracted. And as you can see in the image here, you have a situation where something like the fingers look like they're contracted due to muscle contraction, but it's really the development of this fibrous tissue. And so the muscle is frozen and it restricts the mobility of joints. And then again, as in the image, you have a situation where the muscles look contracted and you can't move the full range of motion at those joints.